What's good, folkers? Snapzilla Gorilla, aka The Definition, hitting y'all up with another video. It's been a minute. I actually been I figured out a way to do my presentations on the computer using a certain software, but man, with my kids running around and me trying to get some sleep, and work, I didn't really have time to sit at home in front of the computer because the computer is right next to the living room and kids are running around, and there's no way I could really make that happen without, being disturbed by the children. I'd have to wait until at night when they're asleep, and but the next thing I wanted to do was a presentation on viruses, okay? And I'm just gonna go from a tablet, and any images that I need to show you guys, I'll get from the tablet, and we'll go from there. All right, let's get into viruses. First, let's see what they are. Now, this is not going to be a long-winded, every aspect of this presentation is going to be cut and dry. I ain't got time to play. Yeah, so let's do this. This is YouTube, by the way. This is I'm just giving a little information about viruses that I found, okay? I'm not making any assertions. I'm just giving the people what I found. All right, so as you guys know, when I do my presentations, I'm going to give you links in the description to the sources that I have so that you guys can go and check it out for yourself, read the whole thing if you feel like it. If not, that's cool too, So what are viruses? Real quick. Let's get into it. Viruses are microscopic parasites, okay, according to Live Science, an article in Live Science, okay? Yeah. So, viruses are microscopic parasites, generally much smaller than bacteria. They lack the capacity to reproduce and thrive outside of a host body. They lack the capacity to thrive and reproduce outside of a host body. Interesting, right? All right. How much smaller are viruses in comparison to bacteria? Quite a bit. With a diameter of 229 nanometers, the measles virus is 8 times smaller than E. coli. Okay, so basically, viruses can be anywhere from 20 nanometers to like 400 nanometers to even bigger than that. Just many different types of viruses. Okay, now, what role do viruses play? Well, let's look into that. Okay, we know what we've been told, that they cause disease, they infect the cells, blah blah blah. But let's look at let's look at some other roles they might play. All right. Now, this is from the Journal of Molecular Biology. Okay, this next article that I'm reading from. Many viruses. Okay, so the molecular basis and role of viruses in human microbiome. Okay. Highlights. Just so you know, I'm reading straight from the article here. Many viruses are stable members of the human microbiome. Many viruses are stable members of the human microbiome. Viruses of the human microbiome provide evolutionary advantages to some bacteria. Viral biota traverse human mucosal barriers. Okay? That's something that they told us, right? Bacteriophage in the microbiome protect mucosal barriers from potential pathogens. Bacteriophage in the microbiome protect mucosal barriers from potential pathogens. Viral biota may be involved in the dysbiosis that results in human disease. Maybe. Okay? Now, here again, viruses are dependent biological entities that interact with genetic material of most cells on the planet, including trillions within the human microbiome. Their tremendous diversity renders analysis of human viral communities, known as Veeeruses, to be highly complex. What does it say here? Analysis of human viral communities. What does that mean to you? Not a doorknob community, not a hard surface community. Human viral communities to be highly complex, meaning they don't really know shit about it. They're still learning, okay? Because many of the viruses in humans are bacteriophage, meaning they attack, basically attack bacteria. Their dynamic interactions with their cellular hosts add greatly to the complexities observed in examining human microbial ecosystems. We're only beginning to be able to study human viral communities on a large scale, mostly as a result of recent and continued advancements in sequencing bioinformatic technologies. Bacteriophage community diversity in humans not only is inexorably linked to the diversity of the cellular host, but also is due to their rapid evolution. What does that sound like to you? These things help people evolve. Horizontal gene transfers and intimate interactions with host nucleic acid. There are vast numbers of observed viral genotypes on many body surfaces studied. I mean, look. Now, let's get more into what the role of a virus can be, okay? And I'm going to show you a little image here. Hopefully it works out. Hopefully you guys can see it good. So, what are we hearing about viruses all the time? That they're bad, right? They're killers. They infect us. They give us diseases and whatnot, okay? But let's look at this. This is a little different. This is from KQED, okay? It's another scientific journal.org. KQED.org. I got an appointment, so I'm making sure things are okay. So, there has been a lot of news lately about bacteria living in our gut, the human gut microbiome. Researchers are learning which bacteria live there, who is naughty and who is nice, and even somewhat distasteful. Way to replace naughty with nice. A fecal transplant. Okay. What gets lost in all of this is the fact that bacteria are wild creatures whose only purpose is to divide and spread. If given half a chance, even those helpful bacteria would break free of the gut and spread through the body, eventually killing the host. Right? To properly harness their power of good, to properly harness their power for good, meaning the good bacteria, we need to have ways to control them. A new study presents an unexpected way that we work with viruses to keep bacteria confined in the gut. The idea is based on the fact that the mucus which lines the inside of our gut, is riddled with viruses already. Riddled with viruses. The mucous membrane in your gut is already filled with viruses. Okay? The idea, so if bacterium gets too far into this mucus layer, it meets with a phage that destroys it, the virus. It is like a minefield of viruses set to explode whenever they encounter bacterium. Except that it is even better than that. It's a self-renewing minefield. When a phage infects a bacterium, it first hijacks the bacterium cellular machinery and forces it to crank out lots of new virus particles. This is what we've been told viruses do to human cells. Once a bacterium has made as many virus particles as it can, the bacterium then explodes, releasing lots more phage into its surroundings. Now there are more phage that can infect any bacteria bold enough to venture where they shouldn't. So, as you can see here, viruses are instrumental in keeping your gut in check, right? That's what it's saying here. Research has been done. Either way. Okay? Obviously, viruses keep bacteria in check inside the gut. And it uses the cell, or it uses the bacteria to replicate itself. Okay? But I want y'all to pay attention to this. Now, I've shown a picture of it. Now, this is the virus that you would see in the gut. Okay? Bacterial viruses, phage, like many, form a shield to protect us from living bacteria living in our gut. Image on the left is a scanning electron micrograph of one of the phages. And the image on the right is a diagram, okay, of a similar phage. So this is an actual image of a virus, right, that attacks bacteria. This is a bacteriophage that keeps bacteria in check. Your body uses these things to keep the balance in your gut on point. What was it? Now, this is just one of many types of viruses. I'm just letting you know. Our body uses viruses, and not only that, the viruses only can exist within our bodies, right? I said it mentioned it in another article before. Okay, now it might be in the skin and stuff like that, but once it's separated from that, you need, like I said, it needs a host. It needs bacteria, it needs whatever. Okay, so this is what I'm talking about here. This is just me giving you guys information. And YouTube, this is being fact-checked. The things that I'm saying have been fact-checked. I will present links in the description. So, I mean, I'm not monetized anyway, so it doesn't really matter right now. But, it is what it is. All right. Now, let's get into, real quick, exosomes. Okay? I don't know if you guys have heard of those. Those are also things in the body that are roughly the same size. It can be the same size as a virus. And there are many different types, like virus, that your body creates, or that perform different functions for different cells, things like that. Excuse me. So what is an exosome? Let's see. An exosome is a subcategory of bilayer membrane-bound nanovesicles released from normal and disease cells into interstitial spaces, and in some cases into bodily fluids. Exosomes are also cultured by cells into media, meaning that they'll put exosomes onto some type of flesh or something and see how they interact with it in a petri dish and take images. That's what that means. Exosomes are defined and separated from other vesicles based on their source, method of isolation, sizes, and surface markers. Surface markers, remember that. Exosomes and other vesicles frequently are unrecognized as to their importance to physiological and pathological processes because they are essentially invisible. Their small size keep them suspended in fluids so that their effects may not be identified, in that their contents and functions would seem to be consistent with those of soluble molecules. So, exosomes and related microvesicles were once thought to be artifacts and or cellular trash, but now exosomes are accepted as a component of newly identified intercellular communication system that can modulate the functions of target cells. What do viruses do? Basically the same thing. All right. Now, what is the size? So a subgroup of such microvesicles as exosomes is about 30 to 100 nanometers, okay, in size. All right, that's just certain types. All right, similar size to viruses, okay? Now, I'm not making any assertions. I'm just pointing things out. I'm not a doctor. I'm not a scientist. I'm just going by what I read. I can read and I can comprehend, and I do have a little bit of common sense. So, It is what it is. I'm just giving you guys what I'm reading here. All right. So, now, I am going to show you guys. I thought I had an image of a. All right, here we go. Let me add it here. Bear with me. Exosome. I had an image of exosome up. I don't know what happened to it. I'm only interested. I'm gonna show you guys some images, and I'm only interested in images of actual pictures. I don't want to see no freaking CGI picture. So I just wanted to show you guys a picture of a virus. So, damn it. Sorry, guys. I want to show you an image of a virus versus an exosome. So this is a picture of an exosome. This is the cell, and this is all the shit inside of the cell. That's the exosome, okay? And it's coming out of. Hold on, let me see. Okay, so a transmission electron micrograph of a cancerous B cell that has secreted exosomes. Secreted. Came out of. See? Now, I'm going to show you a picture of a virus. Right? This is supposed to be coronavirus. You know, this is colorized and all that. But I want you to pay attention to how similar they look. Not to say that they're the same thing. I'm just saying. But even beyond that, I want to point something out. Now, I've Googled as many images as I could of an actual image of a virus. And I tried to find pictures of them entering a cell. I can never find it. I can only find pictures of viruses coming out of a cell or emerging from a cell. Never seen them going in. Never saw a picture of them going into a cell. All right. Now look. Right here. At the bottom. See, this is an image of viruses. Virus particles are shown emerging from the surface of cells in a lab. Emerging from. That's what it says at the bottom. Okay? This isn't the only website that shows these. So, and the caption here says, the scanning electron microscope image shows SARS-CoV-2, which is the coronavirus, isolated from a patient in the U.S., emerging from the surface of cells. Emerging from, coming out of the cell. Not going into, coming out of. I have yet to find an image, a real image. Now this is colorized, but it's a real image of viruses entering a cell. I have yet to find it. If anybody can find it, please put it in the comment section. I don't think I'm right about everything. I don't think I know everything. But if you can find it, let me know. Those are just two examples. I'm not going to go through every single one. I saw plenty of pictures of viruses exiting a cell, emerging from a cell, never going into one. All right? I'm just saying, YouTube. Just saying, I'm not making any assertions here. So, and with that in mind, how do they test for these things? How do they test for viruses? Well, just so happened I researched that too. And I went to many different places, and I just wanted to find the one that breaks it down pretty well. and I'll put all of them in the description. All right. There are several types of tests. There is a rapid molecular assay, rapid immunofluorescence, viral culture, serologic testing, novel influenza A virus infections. Oh, wait, no, my bad. Now, this is from the CDC, okay? Let's go through a little bit of each type of test that they do. So, let's start with this. Rapid molecular assays. Rapid molecular assays are the kind of molecular influenza diagnostic tests that to detect influenza virus nucleic acids in upper respiratory tract specimens with high sensitivity and specificity. My bad. The FDA cleared rapid molecular assays available. So basically, they're looking for markers of a virus when they do the test. Okay, markers of a virus. That's what they do. So they'll perform some type of chemical chain reaction with your cell, and if it secretes some type of nucleic acid, they say, then that's how they know you have the virus. All right. So another type of molecular assay is reverse transcription polymerase chain reaction. Okay, so yeah. So basically what they're doing is they're basically, and in some tests, which one was that? So basically, with this chemical chain reaction thing, they're doing something to the cell to get the cell to release some type of like they said, nucleic acid. Okay. Identify the presence of influenza viral RNA or nucleic acid in respiratory specimens. You know. But my question is, how do they get images of viruses emerging from the cell themselves? See, that's my problem here. If the test they're performing gets the virus to come out, or get the markers to come out, I mean, what did they do to that cell to make that happen? And would that happen without that happening to that cell? Is there some type of... could it be that something is happening to the cell to send it into distress to release a virus? Could it be that? I'm not making any research. I'm just asking the question. Could it be that when they perform these tests, either the cell is going to or not going to release the viruses depending on how under distress it is? Or is the cell already under distress when the sample is taken, right? Either by industrial factors or chemical factors like the shit we breathe in the air, or the shit we put on our skin, or the food we eat, or the injections we get. Are they already going to be under distress? And so when you take the sample, your cell's already under distress, and then you put some chemical shit on it, and then boom, here we go with viruses coming out. Why can't we ever get an image of viruses going into a cell? I don't know, man. Y'all tell me. Well, I'll tell you what. Let's do some more on the testing. Tests are turning up negative. Okay, this is about... I'm going to get into a little bit of the coronavirus testing, okay? Coronavirus tests are being fast-tracked by FDA, but it's unclear how accurate they are. Okay? And this is from Prepublica. All right? So, it's a non-profit newsroom that investigates abuses of power. Sounds like my cup of tea. So basically, they're saying that 30% of the tests that are being given are coming up with false negatives. Okay? Now, I tell you what, man. I tried to find. So, what is the rate of false positives? Let's see. What? False positive. for that cerveza bug. Let's see. Y'all bear with me. All right. Let's see. Toward data science. All right. Okay, so yeah. Basically, there are a lot of false negatives, false positives coming out with these tests. And I want you to understand something very important about when it's a false negative. Okay, when it's a false negative, as outlined in this this article here, doctors are compelled to say, oh, well, you're showing all the signs and symptoms of having the coronavirus, so it must be the coronavirus. So they're still insisting on diagnosing people with coronavirus. But my question is, has something else ever caused us to have respiratory issues like that? Now, I saw a video, I heard a video my brother was playing that was talking, it was a doctor talking about how in New York, the way that they're using the ventilators, okay, the way they're being told to use the ventilators are basically for people who can't breathe, the muscles around their lungs aren't doing the voluntary process of compressing and relaxing the lungs, you know, helping the lungs do its thing, contracting and relaxing the lungs, right? So there's different ways you can set up the ventilator to perform, okay? And they're saying, this doctor said that they're being instructed to set up the ventilator to where it's hooked up to a patient who can't breathe on their own, when really it's more like altitude sickness, where you're not getting the proper amount of oxygen, or you're not getting the proper amount of oxygen in your lungs. So like when you're at very high altitudes, like say you're at Mount Everest, and even likened it to dropping somebody off at the top of Mount Everest, right? Just straight there. That's more of what it's like for these patients that they end up putting on these ventilators, okay? And this is why they turn blue. This is why their skin and their lips turn blue, because it's not because their lungs won't allow them to breathe in and out. It's because the quality, something is preventing the quality of oxygen, or preventing the proper amount of oxygen or good quality oxygen from reaching the bloodstream to be attached to the hemoglobin to be taken to different organs that need it, like your brain, your heart, things like that. So, interesting stuff. No assertions, YouTube. I'm just saying. All right, so that's pretty much all I got for now, y'all. Hope you enjoyed the video. Hit me up in the comment section. Let me know if you can find an image of, or something of, showing a cell or a virus going into a cell, and then coming out. That's all I want to see. That's all I want to see. Anyway, much love to y'all. Like I said, man, I'm gonna try to get on the computer and start doing more of this type of shit. I found a way. I know how everything's set up. I know how I want to do everything. Is just getting my shit together. You know, that's all it is. Just getting my shit together to make sure it's working properly. You know, make sure that, you know, I'm not going to be disturbed. Make sure I can go through the presentation without many issues. Anyway, much love to y'all. I'll holler at y'all later. Y'all stay safe out there. And I'm going to come at y'all later about a lot of hypocrisy. I can't even talk today. A lot of hypocritical BS that's going on with our government and these functionaries and these executive officers known as the police. It's getting wild, y'all. It's getting wild. It's time to make something happen. It's time to make something crack. It's time for, I don't know, maybe something. Anyway, I'll let y'all later.